and intracellular protein degradation in eukaryotic system. I repeat, we are talking about the intracellular protein degradation in eukaryotic system, and the major property of this system is selectively degradation process. So the process is again selective, very selective. Remember, I have told you the specificity, and also you can say selective. So specific and selective is a little bit they are different in literary, but in this case stand for a very close meaning. Okay, so they are very specific and selective for their degradation purpose. Now let's talk about the beginning of the proteolysis process using the ubiquitination pathway. Now the process of ubiquitination here, it has three different steps. One is the covalent modification of the target protein. So it's not like that you get only target protein and degradation just happen like that. It is a very specific process. It's selective, and also it's low, as we have already discussed, SSP, and obviously it's a processive type of processing. That means the process will occur step by step and slowly. That's why we call it a processive, like it's like a processing of the polypeptide chain. So first thing is to modify this target protein, and the modification brought about is covalent nature. So that's why it comes to another type of important assumption, is that all of the processes that we are looking in case of this proteolysis is irreversible, right? Sorry for my bad handwriting, but that's another very important assumption, that these processes are irreversible in nature. Okay, because we are having a covalent modification at the very first step of it. Once it is modified, and no longer it can be reversed back to the previous case. Second thing is to unfold those proteins, because the machinery or the actual giant reactor which will degrade those polypeptide chains are called a proteasome, right? And this process is done in collaboration of two different things. One is the ubiquitin, second one is proteasome. One is ubiquitin are small molecules. We are going to talk about ubiquitin later in huge amount and in detail. We'll be talking about proteasome in detail future, but this is a collaboration of two different systems together, ubiquitination as well as proteasome degradation. So first thing is to covalent modification of the target proteins, second is the unfolding of this protein, and third one is the degradation of those proteins by 26S proteasome complex. Okay, now what is proteasome, what is ubiquitin? Let's find out. Now here it is, the structure of a ubiquitin molecule. Now ubiquitin is a 73 or 76 amino acid long protein polypeptide chain. So it's a pretty small polypeptide chain. It is having a structure mostly made up with, it's a blend like made up with mostly of beta strands, as you can see, beta sheets, and a little bit of alpha helix, and also the most important part of this structure is this glycine tail, which is present in the C terminal. So the C terminal glycine tail for this ubiquitin is unique, and it is important for the tagging, because what is important in nature is that if we go back and look at the overview of protein degradation, which we haven't talked about, is something like that. We have these ubiquitin molecules, and what these ubiquitins are doing is they just tag those proteins. They just tag and hold on to the target proteins, and for this tagging they must require the signal or degrons, right? So these proteins are having degrons. So suppose this green one is a target protein, here it is having degron, and these ubiquitins are being targeted to these degrons in a ubiquitin mediated pathway. We'll be talking it later. So simply those target proteins are tagged with polyubiquitin or many ubiquitin molecules. This step is called polyubiquitination of target protein. After the polyubiquitination has taken place, that what will happen, then it will be targeted to the proteasome, and when it will be targeted to the proteasome, utilizing ATP, proteasome will chop those polypeptide segments out into small peptides like 15 to 18 or 20 peptides long. Okay, so that's the overview of this method of proteasome mediated lysis. Okay, so here it is. So we know that these ubiquitin molecules are required, and polyubiquitination chain formation is also required. So how the polyubiquitination chain is made? So now let's look at it in the very close view in the biochemistry of this ubiquitination. Now here it comes. Here it is a substrate. Substrate means here it is the protein or the target protein of our interest. Now we have seen in the picture that suppose this so let me change the color first. So say here it is. Suppose this green thing or this green colored molecule is, where am I drawing? Okay, so this green color molecule is our desired protein, and we need to target these proteins with the ubiquitin molecules. And let's see this red, so let's see these blue things are the ubiquitin molecules. So here comes the ubiquitin molecule. So we need to target several round of this ubiquitin molecule with this protein, and they will be added using this C-terminal glycine tail. So here comes the importance of this C-terminal glycine tail of ubiquitin. Now if we look at the zoomed view, now here comes this ubiquitin with its C-terminal tail, which is the glycine at the C-terminus, and at the N-terminus it is having an NH2 group, which is a common feature of a protein molecule, because this is also a protein. And obviously here it is a substrate, and the important feature of ubiquitin is that ubiquitin molecules all the time, most of the time, targets the lysine residue of the substrate protein or the target protein. Okay, so here it is the substrate or target protein. It is having the lysine residue. We all know that lysine residue is having the NH2 in the R group. So it will target this lysine residue and the R group of the lysine residue, and the R group is having NH2. So again, they will be interacting using this carboxyl group and this amino group, epsilon amino group. So the interaction here with carboxyl group of the glycine that are present in ubiquitin with the epsilon amino group of the or the amino group of R chain of the lysine that are present in the target protein. So after the interaction, what we get, a CONH bond is formed in this case. Okay, so this kind of amide bond is formed. So this is called amide bond. Okay, so this is also called a isopeptide bond, which is getting formed between the ubiquitin and substrate. After this bond formation, this linkage is properly done. Now this is one step of attachment, simply the first attachment of ubiquitin with substrate. But for the further ubiquitins to be attached, the ubiquitins to be attached with rest of the ubiquitin, right? So here first interaction is between the substrate and ubiquitin, and second interaction is ubiquitin and the rest of the interaction will be between ubiquitin and ubiquitin. So the ubiquitin molecules must have another NH2 of its lysine, because remember, and the next ubiquitin, which is the second one, here it is the second ubiquitin, it is going to attach with the first ubiquitin with its glycine tail. So the second ubiquitin is having the glycine tail, it will be attached with the first ubiquitin using the NH2 group of its lysine. So again the interaction will be same, the process will be same. Instead, in this case we are having substrate and ubiquitin. In all other cases we will be having, instead of substrate, we are having an ubiquitin molecule. So ubiquitin molecules are also having lysine residues in them. Using them, these lysine residues, they are attached with the next ubiquitin molecules to form the polyubiquitin chain. Okay, so here it comes about the polyubiquitination, as you can see here. So suppose here the colors are given. So this white region is for the glycine, and this blue region is for the lysine. So here the lysine on the surface of substrate, it is bound with the glycine of the C terminal of first ubiquitin. So this is the first ubiquitin. Now if we look at for the second ubiquitin, the second ubiquitin is having lysine. Here it comes the lysine, blue one, and then the lysine is attached with the third glycine of the third ubiquitin, and so on. We are having the attachment like that. So we must have a lysine somewhere at the top region, must have glycine at the C terminal, right? So that's the important part of all. Okay, so several lysine residues within a protein can be modified at once. Now that's another very important concept. Now the modification that we're talking about here, it is a substrate, and many ubiquitins are tacked onto the substrate from this region. But it may possible that the ubiquitins will start attached from different sections of this substrate. So suppose these substrates are attached to many ubiquitin poly chains from different regions. So multiple modifications can also be possible, or multi-tagging is also possible between the ubiquitin or polyubiquitin chain and also with the substrate. Okay, now here we can see the detailed steps. We are having the E1 with SH. So these are the ubiquitin molecules. There are three different ubiquitin molecules. One is E1, then E2, then E3. Now E1 ubiquitin molecules are very specific, not very specific. They are very less amount. They present in very low concentration inside the cell. On the other hand, E2 and E3 like ubiquitins are present in very high concentration inside the cell. Why? We'll be discussing it in the later slides. Now here it comes and the talk about E1, E2 and E3. ubiquitin molecules. Now, as I've told you that E1 is having a very important job to done. The job is that we need to activate the ubiquitin molecules, because the ubiquitin molecules, when they get synthesized, they present as the unfunctioned form inside the cell. So we need to activate those unfunctioned form inside the cell. So this precursor of ubiquitin is not fully functional. So you need to make those precursor fully functional, and we can make that precursor functional using a kind of this E1 molecule. So E1 is called the activator molecule of ubiquitin. So here comes the E1 with this SH group here. Now what will happen, this is the free ubiquitin molecule. It is really not at all fully functional at this place also, because it is having a CO minus at its end. So it must have a COSH structure. So what we'll do in this case, transfer of this free ubiquitin molecule onto the E1 is done. This is the first step. For this transfer, it requires some energy. The energy is derived from ATP hydrolysis. So ATP is hydrolyzed into AMP, and inorganic pyrophosphate or PPI is getting released. So this is the first step. After that, what will happen? What we get the U1 attached to this free ubiquitin molecule. So ubiquitin is no longer free. The ubiquitin is now attached with the ubiquitin activator E1 molecule. Now here comes the E2 molecule. Now E2 molecule is called the ubiquitin transfer molecule. E1, I said that E1 are activators and E2 are transfer molecules. We need many E2 transfer molecules, and this is going to play a very vital role, because once the ubiquitin molecules are getting activated, we no longer need E1, but we will need E2 to transfer these molecules to another major molecule, which is called E3, which is the most important component above all in this ubiquitination system. Okay, so here it comes. So once we get the ubiquitin molecules attached to E1, they will transfer this molecule onto the E2. Again, the transfer, E2 is also having SH group, so the transfer is from one SH group to another SH group. So it's a simple SH bond, this thiol bond breakage and the same kind of bond formation. So it will result in net change of energy 0, because we don't require any energy of breaking and making same type of bonds. Then what we get, we transfer this ubiquitin to the E2. So now this ubiquitin is ready to be transferred and ready to function properly. So this is the ubiquitination activation phase, until and unless we transfer the free ubiquitin onto the E2 molecule. Now one thing you can ask, that's why not this free ubiquitin directly ends up adding with this E2? That actually cannot happen, because E1 is having a separate type of structure and configuration, that it can only activate this E1. So this ubiquitin, so the ubiquitin must be attached to E1 at the first time, then it will be transferred to the E2. So we get an activated ubiquitin here. Now what will be done, this activated ubiquitin from this E2 will be transferred to E3. So that's very important. So now it comes the major part, which is the E3 protein, E3 molecule. Now E3 molecule is called the ubiquitin ligase molecule. So what are the function of ligase molecule? If you know the basic molecular biology, you must understand the ligase is having a job of attachment, right? So it ligates or attaches different segments. So again in this case also E3 is functioning as a ligase. So it is having the two different binding regions. One is the binding regions for this E2 with ubiquitin, another binding region is for attachment of target protein. So this E3 molecule is doing a very great job. So it is having two binding sites. Here it comes a binding site for E2 with the ubiquitin, here comes a binding site of the substrate of the target protein. Okay, so what it will do now, it will bring this target protein to close proximity with this ubiquitin molecule, with the ubiquitin carrier. Ubiquitin cannot transfer on its own, it must require a carrier. That's why it will be attached to the E2 throughout the time. So here it comes, this E3 ligase molecules actually bring both of this target protein as well as the ubiquitin in close proximity, so that this target region of the protein can be attached with ubiquitin molecules. So here it comes the NH2 region of the target protein from its lysine residue, and obviously we know that ubiquitins are having a C-terminal glycine. So here it is a C-terminal of ubiquitin having the glycine residue. So using this C-terminal glycine residue, this ubiquitin will be attached to this N-terminal of or NH2 group of epsilon or epsilon NH2 group of lysine residue of the target via the amide linkage. So amide linkage will be formed, and then this E2 will release the place. So it will be released. Now what will come? Another E2 will come holding another ubiquitin, and so on. The ubiquitin will be start adding to get a polyubiquitin chain like that. Now we know that we need to mark at least 8 to 10 polyubiquitin chain for a protein to be targeted for the proteasome mediated proteolysis pathway. So we need to add at least 8 different ubiquitin molecules together, right? So that's the overall process of activation and then the attaching of ubiquitin to the target molecules. It's called the polyubiquitination of any kind of protein. Now here we've seen a very basic level of E3 ligase, but there are some other kind of E3 ligase which are having three different sections. Uh, one is for binding with the E2 with ubiquitin, another one is for binding the target protein, but it can also bind with raw ubiquitin molecules. Let's see that. So here it comes. Some E3 attach with the ubiquitin also. So here it comes. It is also having an SH domain coming out from it. Now what will be done here? Rest of the part remains the same. So the ubiquitin in the previous case what we have seen, it is directly transferred to the NH2 group of the target protein. But instead in this case it won't be directly transferred to the NH2. Instead it will be transferred to the SH region via amide linkage first. Then what will happen? Then it will be transferred to the actual region. So this thing can happen. So it will vary from species to species and different organisms that we find this kind of things. And another important thing I must tell you, it's a simple name nomenclature region, is that proteins in the other major class of E3 has a single subunit with a segment called Hecht domain. So this is called the Hecht. Okay, so the Hecht class act as direct carriers of the activated ubiquitin. Okay, so for the Hecht E3s, the activated ubiquitin is transferred from the E2 to a conserved cysteine site. So we have seen in this case, and then it will be further transferred to the actual. So this domain is called the Hecht domain. So this kind of ubiquitin ligase is completely different. They are not like the conventional ones that we have already discussed. Okay. So this kind of ubiquitin transfer is called a bucket bridge like ubiquitin transfer. It's simply the naming. You don't need to memorize all these things. Now let us talk about a little bit about the structure of E3, which is the ubiquitin ligase, right? So there's the E3 is having a structure which is also conserved structure throughout the evolution we are going to see. And obviously, if you look at the concentration of different E3 molecules inside a cytoplasm of a cell, you are going to find a very few E1 molecules, but we are going to find many more E2 molecules, and you are going to find very high concentration of E3 molecules, because these E3 molecules are varying in their structure. They are varying in different domain regions so that they can bind and attach with many different types of substrates altogether. So it increases the specificity, it increases the selectivity of different proteins. So that's why they need to design many more type of E3 and E2 molecules so that the degree of freedom choosing different molecules get increased, right? So what is the structure here? As you can see, this is a three-dimensional structure having different regions, as you can see. Now in this different regions you can see the fold. Now this kind of typical fold that we are looking at here, it is called a ring finger domain. A ring finger, it's a modification of zinc finger. Simply it's also a type of zinc finger having a ring like structure. That's why we call it a ring finger domain. Now here it comes. We're having these yellow dots in three different regions. It represents the cysteine residues, and obviously this dot is histidine. Most of the part is it is surrounded by cysteine residues in four directions. At the middle we are having a zinc sit on it. It is called the zinc finger domain, a typical zinc finger domain, and it is forming a structure like the folding is like a ring, so it's called the ring finger. Now if you look at the structure here, it is pretty much clear that the structure is formed in such a way. It is having two different regions clearly. One is the substrate binding subunit here, other one is this E2 with ubiquitin binding region, and this is a part of the ring finger. This red region is a part of the ring finger. So this ring finger region is responsible for the attachment with the E2 with the ubiquitin protein, that we all know that. Okay, now another important assumption in this case I must tell you that this substrate binding region can be varied from one E3 ligase to another E3 ligase, that will be varying, and this variation in the substrate binding regions between different E3s are telling us that there are different regions of specificity of this E3 binding with the substrates. Okay, so here it says the binding specificity, and we have seen also the different substrate binding units can be present. So they are having different combinations of substrate binding units, but the ring finger domains or ring finger regions approximately relies the same way. It lies the same way. Okay, so The similar architecture but different substrate specificity can be there for that purpose. Now, what we get, these variable subunits not only provide sites for binding different substrates, but also provides gap for a different size. So we get, suppose in these three models that what we can find, in this case we get the typical type of distance between these two regions, these two domains. One is the substrate binding, another one is the ring finger domain. In this case it's slightly different, in this case it is much more wider. So this region is called gap region. Get, this region is called gap. So this gap region is also important for the accumulation of substrate of the target protein. So this gap region is also varying depending upon the substitution of the substrate binding regions onto this E3 molecule. Okay, I don't know whether it is, yeah, okay. Now, the recognition of the degron by the E3. We have talked about degron. Now, what is degron? We have already known that degron is a signal which will tell that this E3, E1, E2, E3 complexes and the ubiquitination and the proteasome complex to target a protein for the degradation. So it's a signal. It's nothing but signal. Now, the signal can be phosphorylation. The signal can be any kind of like methylation and glycosylation onto the protein surface. It can be the popping up out of hydrophobic residues outside of a protein that can also send a signal to be of that protein to be degraded. Okay, so by far the greatest degree of molecular complexity is carried out by these E3s, but we have also seen there is a variation in E3 E2. So not only we get multiple variations of only E2, multiple variations only E3, but we are having a combination of E2 and E3. So it is giving us much more combinations, so that's why you can tell it give us much more specificity. So here it comes about the diagram. So this is the protein having this particular sequence, as you can see, DS, GL, DS and all the sequences that can signal us, and the signaling molecule of this via the signaling pathway phosphorylation can be taken place. Now phosphorylation plays an important role to tag proteins to be degraded. Now here it comes, it degrades, it tag the phosphate groups onto this ACE and subunits here. As a result of this tagging with, of this phosphate to this S position, what will be done is that modification will be taken place, and as a result of this modification they are going to be attached to the E3 ligase with the substrate binding regions, and then the ubiquitins will be transferred one upon another, one onto another onto the substrate, and then polyubiquitination will be taking place. Then the proteasome complex will degrade this polyubiquitinated protein. So this is a process. Okay, so recognition of the degron by an E3 is very very important. It is this process is achieved via two complexes. One is the substrate recognition domain, and another one is the degradation signal of the domain. So here it comes the degradation signal, and here it comes the substrate recognition domain of E3. So we can see the E3 plays a very very vital role because it not only binds with the substrate but also it recognizes this degron signal is very important. Otherwise, if it receives good proteins, if it receives the proteins we don't require to be degraded, then it will create a hell of a mess, right? Now there are some degrons that can also found which are hidden. Like we have already talked about the hydrophobic residues as the degron that can come outside, but also we can see some hidden degrons. So as you can see in this picture that sometimes so these are the hydrophobic surfaces. If they come outside, they can be tagged with the ubiquitin ligase, or E3 can easily recognize them and easily ubiquitinate them. But sometimes what they can do, several ubiquitin molecules like that which are having hydrophobic surfaces coming out can be bound or attracted with each other because using the hydrophobic interaction. We all know that hydrophobic interaction is a very good molecular interaction, so they will be forming a bond like that. So it's a dimer formation will be done. As a result, the hydrophobic regions for both the proteins will be gone. In this case, they won't be recognized by E3, so they'll survive for a longer period of time inside a cell. So this thing can also happen. Okay, but there are different levels of complex type of degrons and complex level of degradation processes. So for example, what we get in yeast inhibitor, or in yeast what we get a several lines of degrons, and say 8, 10 or 11 sequence or 9 sequence of degrons, and the degradation can only be taken place if and only if at least 6 of the 9 potential sites of this inhibitor must be phosphorylated. So this kind of tight control can also be found in different levels of this proteasome degradation, because we don't want our valuable proteins to be degraded just like that. So we can see here in this picture for this yeast protein, that this is a signaling molecule which is called the destruction box. Now this destruction box is having a several lines of signals. Now this signaling are prone to be signaling with ubiquitin molecules. Now the signaling can be carried out by ubiquitin molecules, and if and only if these regions are phosphorylated, if the only if among this 9 region, if at least 6 residues are getting phosphorylated, then only the protein will be degraded. So by doing this, what they are providing us, the much more control, tighter control over this process. So we can by looking at these examples, we can tell how much responsible this process is, how much important this process is. So that's why it is much more tightly regulated. And obviously, let's talk about another important concept, or N-end rule. Now, what is N-end rule? N-end is a simply N-terminal of a protein. So here it comes, the N-terminal of a protein. It was seen that if there are some amino acid, if they are present in N-terminal, the stabilization of protein gets increased. On the other hand, if there are some amino acid present at the N-terminal, it will those proteins having a particular type of amino acid at the N-terminal are much prone to proteasome mediated proteolysis. So here it comes the name of this amino acid: alanine, proline, cysteine, serine, glycine, threonine, methionine, and valine. This type of amino acid, if they present in the N-terminal position, it increases the stability of those proteins about or approximately greater than 20 hours. So the half-life of the proteins are increased over 20 hours if they use this kind of amino acid at their N-terminal region. And intrinsically, if there are this kind of proteins like arginine, lysine, histidine, phenylalanine, isoleucine, tryptophan, all these proteins present, in those cases the half-life of the protein is coming from 2 to 30 minutes only. Okay, so it's a destabilizing residues. They are called the destabilizing or intrinsically destabilizing. But there are also some residues called destabilizing residues like asparagine or aspartic acid, glutamine or glutamic acid. If these residues present at the N-terminal end of a molecule, it's not that if they present the whole protein. We're not talking about the presence of these amino acids throughout the protein, but we're talking about the presence of this protein at the N-terminal end of the protein. Now this asparagine or aspartic acid or glutamine or glutamic acid, if they present at the N-terminal and more amount, then this signifies, this tells the protein to be much more destabilizing. And if this alanine, proline, these are present at N-terminal, it gets a much more stabilization for that protein. It's a simple observation. Okay.